Good morning, guys. I am twofold here. I'm slight late start, well, sort of. It was an early start because it's my bestie, Miss McCann's birthday today. And so I had to go to her house really early this morning so that I could decorate her apartment and her car and embarrass her a ton. But I just got back from that 30 seconds ago, and I grabbed our FRQ that we were going to do this morning, which we are, but I think it's too much to copy down today. There's just too many diagrams and it's really wordy, so I think I'm just going to send it to you. It's going to be much easier for you to tackle. So I just, let's see, I need more. So with that said, I'm just going to wait like 2 more minutes to see if anyone else comes, so I only have to send it once. Let's see, T's FRQ U4.3. Is that what we're doing today? No, we're doing 4.4. Nope, we're doing 4.3. Wait, are we? Or is that the one I assigned for homework? Shoot. Oh, nope, that's okay. Okay, great. What about this one? Oh, nuts, that's the one I assigned for homework. Okay, so not that one. Let's do 4.4. Yep, fine. Great. Yeah, still big enough that I think I'm going to send it instead. Let's just make sure this is downloaded. 4.4. Okay, anybody new show up? Okay, nobody in the waiting room. Oh, we got a small little crew this morning. What is it, the gloomy weather maybe? People need to stay cozy or what? Okay. Yeah, what do we have? 4.4. Okay, gals, I just sent you the PDF of the one we're going to do this morning. Just be easier for you to be able to see the diagrams rather than try to copy them off my screen. I am also going to share my screen right now so that you can take a gander and make sure that it matches in case somehow in the download or the sending it didn't work. It was so weird a couple weeks ago. It didn't I'll go all the way through. Okay, so here we are. It is a long FRQ. For some reason, it looks like it's missing part B. I don't know how that is. Whatever. We'll do A, C, and D today. No problem. And you can just get that downloaded. I'm going to read through it all the way first, and then we'll just talk about some of the task words that they give you. Again, those task words are things like describe, explain. Now I can't think of anything besides that. Describe, explain, discuss, graph, things like that. Those are all task words. Okay, so this one says, the p53 protein is a transcription factor that regulates a cell's progression through the cell cycle after DNA is damaged by UV light. The p53 pathway is shown in figure 1. Oh, look at how delightful. Oh, don't we just love us some p53? Okay, figure 1, it shows that the role of p53 is regulating cell cycle progression in response to DNA damage. Perfect. We know all about this. Okay, question A. A skin cell completes one round of the cell cycle. Describe the products. Okay, B we're not doing clearly because it just does not exist. C says, draw an X on the template in the space provided for your response to indicate the phase during which the replication of damaged DNA would occur. So if the DNA got damaged, where would it occur? And all you have to do is draw an X. And then D, based on figure 1, way back up here at the top, based on figure 1, explain how a mutation to p53 may lead to increased risk of cancer. Okay, we've got to be experts on this, right? We're in good shape. This is a lovely question. Let's just kind of go through it for some of the task words just to be sure. So in part A, we see that it says describe. I'm not going to go to a notes page today because I think that we've, not that it's overkill, but writing it over and over again, I think we're in good shape. I can just talk through it. I wish I could annotate this. Can I annotate this? Let's just try something. Let's just see what happens. Let's just see. Okay. Can you still see the problem now that I opened it as a PDF? Sadie, will you give me a thumbs up? Do you still see? Oh, okay, good. Okay. Okay. Now, interesting. Can you see me annotating?
No.
No, you can't see my purple?
Mm-mm.
Oh, that is just too much of a bummer. Really? Your screen sharing is paused. Why is it paused? Resume share.
Oh, no.
Okay, let's try this. Stop share. Okay, let's try this again. What about this? What about this? Okay, you can see that, right?
Yeah.
Okay, and then what about if we, how about that?
Yeah.
How about that?
Yeah.
Oh, hooray! Oh my gosh, okay, this is gonna be a game changer, guys. Now I can annotate on this. Great, okay, anyway, back to the FRQ. Okay, so part A says, describe the products, right? Oh, watch, now it's not gonna work, now that I said I could do it. Oh, there we go. Describe the products. So our word is to storytell. So remember that when you describe, you're not only you're not only explaining, right, making it really clear and plain, but you're also storytelling. This is where the detail comes in. So maybe it's easy to remember when D, describe, or discuss, those ones have great detail and they are usually a little bit longer. So this is going to be 2+ sentences, right? I mean, you don't want to go on and on and on and on, but you definitely want to paint a picture for your reader here. Also, anytime they give you a number of anything, just make sure you make a note of it, just in case. It says that a skin cell completes one round of the cell cycle. So what are the products after just one round? Okay, just heading down to part C here. Draw an X. That's it. Draw an X on the template in the space provided for your response to indicate the phase during which the replication of damaged DNA would occur. That's it. Draw an X. Don't do anything else. Just put a big old X right where you think it is. Easy peasy. And last one. Based on figure 1, explain, right? Make it clear. Plain, comprehensible. That's not how you spell. Comprehensible. How the mutation to P53 may lead to an increased risk of cancer. So don't go too crazy here. Explain is one less than describe. So maybe 2-3 sentences max. All you're doing is relating P53 to cancer. Okay, I'll turn you loose. It's supposed to be a long FRQ, but since we're missing B, maybe I'll see if I can find B while you start to work on A. But I don't think you're going to need more than 15 minutes for this. So jump in. Ask questions. I'm going to search for part B. See why you gotta be like that when you download? That's just rude. How does that happen? How does when it downloads, it decides to just omit one? I wonder if I cut this, if I can paste it into here. Nope, I want to paste it into here. No, that's not going to be a thing. That's annoying. Well, I guess we could just write it in. Okay, guys, I found B. I don't know if you can still see me sharing. It just didn't download for some reason. Look, here's the doc, and here's the PDF. Just disappeared. Okay, so it's B says, based on figure 1, explain how P53 regulates the cell cycle in the presence of damaged DNA. So it's another explain. So it's a make it clear, make it plain, make it comprehensible. 2-3 sentences max. Explain how P53 regulates the cell cycle when DNA is damaged. So that one's definitely concept-based. And as far as it goes, feel free to look it up if you can't remember. Right? If there's any part of this that isn't ringing a bell to you, maybe it was too long ago or whatever, this is the time to look it up and get it right. Okay, I'm going to throw this into the other doc. Explain how P53 regulates cell cycle. Sadie, will you just give me a thumbs up that you can see B that I wrote in? Oh, maybe you're not actually Sadie. Sadie, are you a picture of Sadie today?
I don't know. I'm using multitasking, so I don't know.
Oh, gotcha. Now it's black. A minute ago it was a picture of your face, but it definitely wasn't moving. And now it's nothing. Every time I talk to you, just we're making the same face, and I'm like, hello?
No, I can see the question.
Okay, good. how we doing? Just checking in. It's only been 10 minutes, so still plenty of time if you need it. Just checking in, seeing if you need anything or have any questions so far. No news is good news. Sadie, yeah, what's up? Oh, you're done. Gotcha.
Oh, I was just checking if it worked again.
Oh, yes, I can see your face.
I'm finished with the
Great. Jayla's
FRQ.
done,
Yeah.
Sadie's done. Michaela, Moira, how are you guys doing?
Yeah, I'm done.
Oh, good. Moira's done too. Okay. Fantastic. Then let's have a chat. I'm going to just share my screen so that we can talk about it while we're looking at it. Perfect. This is great now that I know that we can annotate together. Super. So this was meant to be a long FRQ, meaning you had 25 whole minutes. Obviously, we just came in about 15 minutes, and all three of you are saying, or all four of you, I should say, are saying that you're done and feeling good. So that's encouraging. And don't forget that that's a good sign that when you know the answer and you write it down fast, that's great. Don't feel like, oh man, I should fill my time. I need to write more. It gave me 25 minutes, so I must have to fill all 25 minutes with information. No, 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 no. Because again, you always, 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 wait, I'm gonna write this because I could never write this one enough, but you could always be concise. There's nothing wrong with being concise. All that means is that your reader, your scorer, is not going to have to search for your answer. They're going to see it plain and clear because you knew what you were talking about, and everyone can move on. That is their goal. That is your goal. That it's not something they have to hunt for. If they have to hunt for your answer or kind of try to figure out what you mean by that, that's going to mean the score is going to start to drop. Okay, so the faster they can whip through it and see you know what you know, the better. Okay, with that said, let's just look at A and talk about that. Here's some answers, maybe ask some questions. It says a skin cell completes one round of the cell cycle. Describe the product. So what's the cell going to produce after one round of the cell cycle, assuming that figure 1 is what just happened. We've got some DNA damage by UV radiation. Anyone want to share?
Wait, I thought that we had to. I thought that wait, do we have to answer part A based on the diagram, or just in general, like if a skin cell completes one round of the cell cycle?
Now that you're saying that, that's a good question. I took it to mean from this diagram.
I did the
I
same thing
just-
as Jayla.
You
I
took it
did
as
it like
from one round.
general.
Yeah.
okay, you took it in general. Sadie, you took it as in general as well. Moira, how about you?
I took it in general.
Okay, then that's on me. Okay, then let's go with that. Maybe I misinterpreted that. You're right, it doesn't say anywhere based on figure 1, so just in general. A skin cell completes one round of the cell cycle. Describe the products. Yeah, I'm with you guys. Okay, so does someone want to share what they wrote then?
Sure. Mine is super short.
Okay.
After one round of the cell cycle, one skin cell would produce 2 genetically identical daughter cells. These daughter cells have a diploid set of chromosomes with the same genes as the parent.
Okay, great. Anybody else?
I said the same thing.
Same thing, okay. Anybody else? Something different? Okay, I'm just writing myself a note here that we used a cell cycle in terms of a healthy cell cycle. All right, that's fine. I'm just gonna say out loud in case, it makes it more clear that it is figure 1 in a different question or a different version of this question, that had it been, pertaining to figure 1, that after the cell cycle, that you would still have 2 new daughter cells, but they would have passed on that genetic mutation now, or whatever DNA damage occurred, that the DNA damage then would be in the new daughter cells as well. That's it. That's really the only difference. That as the cell replicates, that problem goes along with it. It keeps passing on the problem. So yeah, that's pretty much the only difference for that one. But otherwise, those sound good. This is a great place for maybe a quick diagram for your reader too. Maybe you had, drawn the one cell and just shown, whatever it has, 2 sets of chromosomes or something like that, and that, by a full cell cycle, you end up with 2 new ones. Something like that, Everybody cool with that? All right, let's go to B then. B is the one that I wrote in for us. It says, explain how P53 regulates the cell cycle in response to DNA damage, according to figure 1. Okay, so that makes sense, I guess, since that we wrote in B, it's saying, now look at figure 1 as if the DNA damage has occurred. So you guys are probably right. So, okay, talk to me about B. Anyone want to share?
I said that P53 regulates the cell cycle in the presence of damaged DNA by binding to the DNA in the nucleus. The DNA then produces cyclin-dependent kinase inhibitor. This inhibits CDK from binding to cyclin and creating mitosis promoting factor so that the cell does not go into mitosis.
Beautiful. That's delightful. Anyone else want to add on or share? Samesies, different?
I pretty much wrote the same thing.
Okay, great. Okay. Doesn't sound like anyone else has anything different, which is great. The only thing I would add to that, Jayla and friends, is that you may want to say that the DNA damage, it signals like a phosphorylation cascade or something like that, a signaling cascade. That protein kinase is the one that starts the activation of P53. Something like that, just at the beginning. Everything after that is perfection. Only because it asks you to refer to figure 1, and we can't get to P53 binding to the DNA without PK1 or whatever protein kinase it is, starting a cascade to activate P53. So P53 itself can't see the DNA damage, or that's not true. It probably could see it, but its engine isn't started until PK1 or whatever protein kinase is out there turns it on. That's the only thing I would add.
So the protein kinase creates, or does it phosphorylate P53 specifically, or does it create a transduction pathway that eventually triggers P53?
It will trigger P53 directly through phosphorylation.
So
Go ahead.
does P53 isn't transcribed and made, like it's just present in the nucleus, and then the kinases activate it to activate the CDK inhibitor?
Correct.
Okay.
Yes. So
Cool.
it's weird. It's like all these funny little proteins and enzymes only have eyes for one thing, where for protein kinases, specific ones are on the lookout for DNA damage, initial DNA damage, and they can basically feel that it's happening. So then they tell P53, hey, start your engine. Now you got to go find it. Protein kinases are on the outside looking in. They can't go find it and fix it. P53 can find it and fix it, but they can only do it if protein kinases tell them to do it. Does that kind of make sense? It's this weird whole loop. I mean, you'd think they'd make it so much easier, right? But no, all the proteins have such a very, very, very specific job. And then once P53 finds the damaged DNA, then it can send the signal outside the nucleus to like a CDK inhibitor to stop the cell cycle at a specific spot. Good questions. All right, are we good on B? Okay. Okay, C. Hopefully you didn't do any more than draw a beautiful X somewhere. Where's your X?
Mine was at the end of the G2 phase.
Okay, I have someone that says theirs is at the end of G2. Sadie's showing me a picture of hers. Hers is right over the S phase.
I
Jayla?
said the S phase.
Okay, Moira, you want to chime in?
Mine's in the middle of the S phase.
Yeah, okay. Yep, so that's correct. It is in the S phase. S stands for? Anyone remember what S stands for? Really? Well, this would have helped you guys for sure. S stands for synthesis. What are we synthesizing? DNA, right? So it asks us to draw an X over the phase during which replication would happen. DNA replication or synthesis happens during the synthesis phase, so S phase. G stands for what?
Isn't
Growth.
that growth?
Yeah, growth. Yep. So G1, we've got growth of all kinds of proteins and things in order to do that synthesis. G2 is all the growth we need and all the production of proteins we'll need to do mitosis. And that G0 is basically indicating, hey, no growth. We're done growing. I just want to exit this whole cycle business. I'm just going to live my best life. So yeah, C should be a big X over the S. Okay, last one. D, based on figure 1, so back at the top, explain how a mutation to P53 may lead to the increased risk of cancer. So it asks you to look at figure 1 and base your response off that, but looking at the cell cycle picture from C as well can also help you a little bit with this. Anyone want to share? I'm guessing you just don't want to share, not that you don't know, because I know you guys know this.
I said a mutation in P53 may lead to an increased risk of cancer because if the DNA is damaged in the cell, I kind of worded it a little weird.
That's
but,
okay.
If the DNA is damaged in the cell, the CDK inhibitor will not be produced. This will allow CDK to bind to cyclin and create MPF, which would allow damaged DNA to enter mitosis and may lead to cancer.
Great. Okay, anyone else want to share? Okay, I'll just go off of Jayla's at first and you can compare your answer to hers. It was really good. She had, she indicated that basically the point is there is DNA damage to the nucleus and that without P53, the CDK can bind and put the cell into continuous cell cycle, meaning that cell can continue to replicate. So words that maybe she could add that would make it more clear that it's going to be this cell that's replicating out of control is just the word, like eternal replication or just, replication will continue or continuous mitosis, anything like that to show that the cell is going to keep doing that with the damaged DNA, right? So you're basically connecting what you said in part A about the fact that a normal cell splits into two daughter cells with exactly replicated DNA. Now you're circling back to that in part D saying, okay, now this cell is still doing that, but with DNA damage every single time, and nothing is telling it to stop because P53 is not active. So that was delightful. Maybe another great picture, time for a picture, or referring back to the diagram in any place is great. Anyone have anything else they added or something totally different? Questions? Okay, so this one, I think the take home for this one, for me, for you, is that, I'm just gonna change my color here so you can see, is that especially in B and D, explain, right, is short. But more than anything, it's really, really, it's plain. So even though you're tempted to use some of the big science word concepts, make sure you make it really clear and easy for your reader. Try to avoid unnecessary steps. Basically, you just want to get to the point that this cell will continue to do cell cycle, or cell cycle is stopped because of P53. Just make it really clear and simple for your reader. And I think that's it. I am curious to look back. Has anyone watched the video that goes with this question yet? No one's jumping
I
up.
think so.
Oh, you have? I know it was assigned already.
Yeah.
I'm
pretty sure I did.
Okay, so maybe that's what is making you guys know that, A was for a healthy cell. Maybe she says that in the video. I don't remember what she said as far as this one goes. But we'll look back on that and just make sure. But good catch on that. That makes sense. All righty. All right. Feeling good? I'd love to hear your feedback about seeing Miss Bird yesterday. Sadie's giving a heart. That's nice. Anyone else have thoughts, feedback?
It was good.
Good. She seemed to be doing well, right? Yeah. Good. All right. Well, I will happily accept FRQs for feedback. I was just wrapping up. I think it was actually both Jayla and Michaela's this morning before I went to Miss McCann. So I'll finish those and send those back to you right now. And I think that's it. Holler if you need me, and I will hopefully see you tomorrow morning. Have a great day.
Thank you.
You got it.
Thank you.
Thank you.